Hello, and welcome back to the Mechanochemistry discussions. This seminar series is hosted by the NSF Center for Mechanical Control of Chemistry, or the CMCC. I'm Ashley Martini. The goal of our seminar series is, as always, to bring the community together. Our seminars stream live on the third Thursday of every month at 11 a.m. Eastern. They're also available to watch anytime on the YouTube channel. I encourage you to check the YouTube channel out because that's where we have posted all of our previous excellent seminar speakers. We also have a great group of speakers planned for the upcoming months, and we hope that you will join us for all of them. A few quick thanks before we get started. First, Dr. James Batiste, the Center Director, Jennifer Belsick, the Center's Administrative Coordinator, and two CMCC students without whom the seminar would not be possible, Katie Floyd and Ryan Kwok. Thank you again for following us as part of this seminar series. Please do continue to do so on the YouTube channel. Lastly, a few quick guidelines. Note that the seminar is being recorded. If you have any questions, you may either post them in YouTube or you can email them to us at cmccdiscussions@gmail.com. Either way, the questions will be propagated to the speaker at the end of the presentation. Last but not least, it's my great pleasure to welcome today's seminar speaker, Dr. Silvina Pagola. Dr. Pagola is a research assistant professor at the Department of Chemistry and Biochemistry of Old Dominion University in Virginia. She was previously a research scientist at the Department of Applied Science at the College of William and Mary, also in Virginia. She graduated with her doctorate in chemical sciences from the National University of Cordoba, Argentina, and has been a postdoctoral research associate at the X3B1 X-ray powder diffraction beamline of Brookhaven National Laboratory, wherein she worked on direct space methods to solve for crystal structures of organics from powders. Her current work focuses on the elucidation of mechanochemical reaction mechanisms in ball mills using in situ Raman spectroscopy, and the application of synchrotron X-ray powder diffraction is planned to start this summer. Her experience related to mechanochemistry started in 2009 at the College of William and Mary, with the mechanochemical synthesis and solid-state characterization of several charge transfer complexes, all towards the elucidation of liquid additive effects leading to co-crystal products of different ionicity. The results obtained with the recently acquired Raman system for in situ monitoring of mechanochemical reaction mechanisms from ball mills will be discussed in today's presentation. It's our pleasure to welcome Dr. Pagola to the seminar series.
Thanks. Welcome, everyone. And I want to thanks again to the organizers for inviting me to present a talk in this prestigious webinar series. Okay, I'm going to speak today on the effects of liquid additives in mechanochemical reactions. So I chose to discuss three types of mechanochemical reactions that I have previously studied. So one of them is electron transfer reactions of the electron donor tetrathiafulvalene, which is this molecule shown here, and electron acceptors, which are derivatives of parabenzoquinone. So in these cases, I will talk about the effects of liquid additives to necessary to obtain single phases of various polymorphs that can be obtained by using different liquid additives. And I will mention some structural analysis from synchrotron X-ray powder diffraction, and I will briefly mention the effect of the polymorphism of the reactant tetrathiafulvalene, different polymorphs of the products. So the second type of reaction to study is a proton-transfer reaction, the formation of a salt between quinine, the antimalarial, and aspirin. And we can see here a proton transfer from the aspirin molecule to the quinine molecule. And in this case, the liquid additives lead to crystalline products, but using no additive, just by neat grinding in a mortar and pestle or neat ball milling, an amorphous powder is obtained. So then I will talk about the characterization of these amorphous phases using infrared spectroscopy and microcrystal electron diffraction. And also about the quick recrystallization of these amorphous under when they are exposed to certain solvent vapors. And the last type of reaction to talk about is covalent mechanochemistry, is a Diels Alder a cycloaddition of the two molecules shown there. And in this case, I will discuss the effects of an additive that was used to modify the rheology of that mixture. So it was possible to measure in situ Raman spectroscopy data to understand, to study the kinetics of these reactions, and eventually in the future, their activation energy by mechanochemistry. Okay, so then what exactly are these charge transfer complexes of tetrathiafulvalene? Well, they are similar to co-crystals, right? But they can have different ionicity or actually charge transfer between the electron donor, tetrathiafulvalene, and the electron acceptors. So they could be fully ionic, so where one electron is transferred between them, or they can be neutral, and they can be fractions also of molecules that are ionized and other fractions that remain neutral. So both donor and acceptor form a radical ions. So tetrathiafulvalene forms this radical cation, and the quinones form radical ions. But it also happens that their charge transfer is not necessarily obvious by a X-ray crystallography. And different polymorphs of the products can have different ionicities. So I have studied various of these systems which are shown there. I have done mostly a polymorphism screening using a series of liquid additives leading to different polymorphs of the charge transfer products. And I will talk about the two most interesting, which are tetrathiafulvalene chloranil that forms these two green and black polymorphs, and tetrathiafulvalene chloranilic acid, which forms these two other polymorphs, which in this case have the same color. And the use of liquid additives was necessary to prepare single phases of the different polymorphs that were used in various cases for crystal structure solution from powders. So the synthesis of tetrathiafulvalene chloranil is shown in here, leading to this green phase, which is pseudo-neutral. So 3 in 10 molecules are ionized and the other 7 remain neutral. And the structure of this material has been known for many years, and it is a low temperature ferroelectric that undergoes a neutral or pseudo-neutral to pseudo-ionic phase transition at around 81 Kelvin temperature. But the other polymorph, the black polymorph, has been only mentioned in literature, and it has been less studied or less well characterized than the green form. So then at the time that we studied these materials with undergraduates, research questions were something like, which are the threshold volumes of the liquid additives that lead to the single phases of the black and the green tetrathiafulvalene chloranil, since often a mixture of them was produced. And do these volumes depend also on the polymorph of the tetrathiafulvalene reactant? So this material, tetrathiafulvalene, the donor, has two polymorphs. One of them is color orange, and it has the crystal structure that is shown there with these stacks of tetrathiafulvalene molecules. And the other one is brown in color, and the structure is shown there, and it does not have stacks of molecules. Okay, so it was seen that that water and dimethyl sulfoxide as liquid additives led to the black form of that was thought to be ionic. So we use sufficiently large quantities of dimethyl sulfoxide as a liquid additive to produce single phases of this material. And we could collect a synchrotron X-ray powder diffraction data at X16C Brookhaven National Laboratory to determine the crystal structure from X-ray powder diffraction. So in here, then, the effect of a liquid additive has been that it allowed to prepare single phases of these poorly studied polymorph, and the phases were sufficiently crystalline to be able to determine the structure from powder diffraction. So the crystal structure confirmed that this material is ionic. So because of the presence of these radical cation dimers, in which two tetrathiafulvalene radical cations are forming these dimers. So the planes of the molecules are very close together, and there is a sigma bond between their orbitals there. So then the stacks of the electron acceptor chloranil were also dimerized. So there is a longer distance between the molecular planes and a shorter distance, and that also indicates the presence of radical anions. So then it was possible to solve this structure and confirm that it's an ionic material. So then, which were, returning to the effect of the liquid additives, which were the effects of a series of solvents or liquids of different polarity towards the formation of these two materials. So we use 12 combinations of both tetrathiafulvalene polymorph, the orange and the brown, with 6 solvents in an increasing polarity or dielectric constant series. So neat grinding produced the green form with pseudo-neutral ionicity, and then low polarity and medium polarity liquids such as toluene, ethyl acetate, and acetone always led to the green form. The high polarity liquid additives such as water, dimethyl sulfoxide, and acetonitrile, sometimes from the orange polymorph of tetrathiafulvalene, produce the black ionic form. So it was also observed that the threshold amounts of solvents that were necessary to obtain single phases of the ionic form were lower for the orange TTF than for the brown TTF. So the reactant polymorphism had also an effect toward producing these two polymorphs of the charge-transfer complex. And acetonitrile only led to the black ionic form from the orange tetrathiafulvalene. So the black form is also isostructural, remains as isostructural down to 20 Kelvin temperature. So the unit cell parameters and distances and angles change very little at 20 Kelvin, and is more dense at the known temperatures, that the temperatures at 40 Kelvin and 20 Kelvin than the green form. For that is also considered the thermodynamically most stable form among those two. So I have interpreted this liquid additive role similar to a kinetic solvent effect. So we can say that the green tetrathiafulvalene chloranyl product is obtained first, typically, and the formation of this material may be under diffusional control. So it's only necessary that those molecules meet, and very easily this material, the green form, is obtained. But to prepare the thermodynamically most stable product, the ionic form, it appears that this the solvent, the polar solvent, is necessary for this to happen, such as water and dimethyl sulfoxide. So these polar solvents may provide shells of of um, uh, of, of solvent molecules that may produce that the ionization of all molecules in that material becomes energetically possible. So it may actually promote the full ionization of all those molecules. Okay, so then the second system that I studied for charge transfer complexes of tetrathiafulvalene is the reaction of the orange TTF with chloranylic acid, which is this molecule shown in there. So instead of having chlorine, now we have two hydroxyl groups in aspara benzoquinone substituent. And using solvents of high polarity, such as water, dimethyl sulfoxide, and dimethyl formamide, we obtain this ionic polymorph of the charge transfer complex. And the structure of this material has been known for a while. It was solved using single crystal X-ray diffraction. And an interesting issue is that solvents of medium polarity, such as these alcohols, methanol, ethanol, isopropanol, and 1-butanol, also produce this ionic form. So, and these liquids have to be used always above certain threshold amounts to obtain single phases of this material. So in the ionic polymorphs, we have a charge and hydrogen disproportionation occurring, in which between two chloranylic acid molecules, one of them has all the hydrogens, so these 4 hydrogen atoms there, and the other one has all the charge, two negative minus two electron, the charge of 2 electrons. And it also has these radical cation dimers. So then this transfer of hydrogens and charge may be facilitated by these protic solvents, the alcohols, that may actually support the transfer, or they may actually exchange hydrogen with chloranylic acid molecules, and those that way support the formation of this ionic polymer. So when low polarity solvents and no solvent are used, then we obtain a new form, which is the same color than the ionic form. Is not distinguishable by color, but its powder diffraction data is different. And this is obtained with heptane, hexane, toluene, ethyl acetate, chloroform, acetone, and acetonitrile that are low to medium polarity additives. So then again, one of those additives was used to prepare single phases, or almost single phases, of this charge transfer complex. And the crystal structure of this material was solved from synchrotron X-ray powder diffraction data. And here is the Rietveld feed. And then it does not contain these radical cation dimers of tetrathiafulvalene, but it contains segregated stacks of molecules, which are shown in this shown here. which are held together by pi-pi interactions. So we also did a neutron powder diffraction to confirm the hydrogen bonding array, because the hydrogens are essentially not visible in general for using X-ray powder diffraction. So then we found that the hydrogens are not are in each chloranil molecule has the same hydrogens as the neutral reactant in this polymer. So then there was no proton transfer. And the materials, by using infrared spectroscopy, the material, it is also a neutral form. It's just a crystal of tetrathiafulvalene and chloranilic acid. So this material also seems only to be obtained by mechanochemistry. It was not obtained by digesting the reactants with the same solvent cereals that were used for kneading or forming pastes with the reactants. So then after this, I decided to study the kinetics of the formation of the green tetrathiafulvalene chloranil form. And we studied that using quantitative phase analysis from a laboratory X-ray powder diffraction. And we determined a second order rate law for the neat grinding. So for grinding in absence of liquid additives. So we measured that, and by forming pastes with diethyl ether, we found that the reaction occurred overall faster, but the kinetic model was different. So this reaction, the formation of the green tetrathiafulvalene chloranil, is I consider it a good candidate for an elementary reaction by mechanochemistry. So since a second order kinetic model may imply that only this material is obtained what only molecules of the electron donor and the electron acceptor meet, forming the products, and there are no barriers for the nucleation or crystallization of the product from the reaction mixture. So using diethyl ether for forming pastes are kneading, so then we found a different kinetic model of the diffusion control type that would be the diffusion of the reactants toward encounters forming the product in across layers of product could be the process that is determining the rates of the reaction under those conditions. So the second type of reaction to study is an acid-base reaction, the formation of a salt between quinine and aspirin. So in this case, using the delta pKa rule, we can calculate those values with software, or we can obtain them experimentally. And indicates is more than 3 delta pKa, so the material should form a salt, and that is observed experimentally, in which a proton from aspirin is transferred to the most basic site of quinine, forming this salt, and the structure is known by single crystal X-ray diffraction. So in this case, now the liquid additives used, various were used, heptane, toluene, ethanol, and water. They seem to support the crystallization of this material. So a crystalline salt is obtained. In all cases, the same crystal structure is shown in using the powder diffraction patterns. So, but using no additive, then we obtain an amorphous mixture by powder diffraction. Okay. So then a research question at the time has been, did this amorphous actually contain a mechanochemical product, or it was just the physical mixture of the reactants? Okay, and it could be obtained by grinding manually in a mortar and pestle, or also by neat ball milling at different milling frequencies. So we studied these amorphous phases by infrared spectroscopy and by microcrystal electron diffraction. So infrared spectroscopy showed that the products ball milled at 20 hertz and 30 hertz, which are these orange and red lines there, did not have a band at 1691 centimeters to the -1, which correspond to aspirin. So aspirin has been consumed and a total reaction, a chemical reaction, mechanochemical reaction has occurred, although the products were not crystalline by powder diffraction. So then one possible explanation for this, a tentative explanation, could be thought from the crystal structure of this material that contains these zigzag chains of quinine and aspirin molecules, which are hydrogen bonded. So looking at those chains from the direction of propagation, that would be this other figure on the right side, we can see that in between chains, there are no really strong intermolecular interactions, such as there is not even hydrogen bonding. There are more like van der Waals types interactions, or weak, presumably weak interactions. So it is possible that those chains have certain degree of freedom to rotate one with respect to each other. It's only is a tentative explanation based on the possible attractive forces in that structure. So then a research question has been, is this amorphous product by powder diffraction also fully amorphous by microcrystal electron diffraction, where we can measure a diffraction pattern from individual crystallized, individual micro particles in the micrometer size. So then these results are shown here, and they were done at Eldico Scientific in Switzerland. These experiments were not done in my laboratory, they were done in Switzerland. And we can see that a scanning transmission electron microscopy micrographs shown there these red zones where the electron beam was also used for diffraction, and we can see that they tested 35 particles, and 2 of those diffracted actually the electron beam. And we can see here these diffracted beams at those dark spots around the central beam. So that means that not all of the particles were really amorphous. Most of them were, but not all. So this is the powder diffraction data. And we can see that there are 2 little peaks, one here peak and one there, but they correspond to the diffraction pattern of aspirin. So there is unreacted aspirin that is left over in that material. So then, which was the structure of these particles that diffracted? Okay, So these two diffracting particles were sufficient to actually determine a unit cell. And the unit cells we can see here determined by microcrystal electron diffraction was the unit cell of the crystalline product that is obtained with liquid additives. So the numbers are very similar. And we can see here the reciprocal lattice by micro-ED. So then it seems that it appears that this amorphous bipowder diffraction actually contains certain fraction of crystalline or poorly crystalline particles of the product. So then other experiment that we did is to expose those amorphous phases to vapors of various solvents that were dimethylformamide, acetonitrile, and hexane. And we can see that using dimethylformamide and acetonitrile, in 1 hour, the amorphous becomes fully crystalline. I'm going to show the diffraction data in another slide. Hexane took a little bit longer, but here I have to measure this again because I have to go home in the middle of the measurement. So this is not the final number there. But if the amorphous is kept under air, it can stay more than 9 months. And the only thing that is seen is the maximum of the diffraction data of the crystalline product that is showing up there a few weeks after it has been left under air. So then definitely these solvent vapors really accelerate the recrystallization of the amorphous. And what is observed is the crystalline product obtained by a mechanochemistry when liquid additives are used. So the unit cell parameters of the recrystallized salt are essentially the same if dimethylformamide vapor is used or hexane vapor is used. So we can see that they are within the experimental error for a laboratory X-ray powder diffraction that was used for their determination. So under the optical microscope, most of the particles still are in less than 3 micrometers size, and they are kind of round shape. And a few particles of larger sizes are observed, but these are really few. The majority of them are these small micrometer size particles. So what happened with the crystallinity of these two powders that were recrystallized with hexane and dimethylformamide? So we did a Williamson-Hall graph, which result is the parameters indicating the crystalline domain size and the strain of those particles of the recrystallized powders. So, and we can see that from the width of the powder diffraction peaks and how that changes with the theta angle in the diffraction data, we can see that the powders exposed to the dimethylformamide lead to larger crystalline domain size than the powders exposed to hexane, which are less crystalline, less good crystallized. So this domain size is smaller. And this could be due, there are many options, but it could be due to the solubility. So like the material is a salt, so it's probably more soluble in a polar solvent such as dimethylformamide than in hexane. And it could also be related to the dielectric constant and the of dimethylformamide, the solvent that should be higher for dimethylformamide. Okay, so the last part of this talk is this one, and more my recent work has focused on studying mechanochemical reactions and try to reuse the homogeneous kinetics and mechanisms knowledge that has been accumulated during centuries of the study of liquids and gases, try to reuse that for the study of mechanochemical reactions as much as possible, especially when they occur under homogeneous type kinetic models. So then this is a recently received system for in situ Raman spectroscopy monitoring of mechanochemical reactions in a ball mill. So the reactions happen in these acrylic jars, which are shown there, and there is an optical fiber probe which goes directly underneath the reaction mixture while it is ball milled. So the system also has a Raman laser that the ones that we have is 785 nanometers. And there is also a Raman spectrometer, and all of this is connected to a computer for measurements. So, so far, we are studying the reaction Diels Alder cycloaddition. So it is these reactions are pericyclic reactions with cyclic transition states and a concerted mechanisms in which electrons move from this double bond and form two other new bonds leading to a 6-atom ring, which is shown there highlighted in yellow. So we know that our product is the product of a Diels Alder cycloaddition because we have the crystal structure. The maleic anhydride has hydrolyzed, so then we have this. We don't have the anhydride. We have 2 carboxylic acid groups, but the 6-carbon ring has been formed, indicating a Diels Alder cycloaddition. So these are the rates that we expect. So this is the conversion fraction of the product obtained versus the time. And we expect that the rate of product formation is equal, but with the sign change of the rate of the decrease in the amount of the diene, the 2,3-dimethyl 1,3-butadiene, and the same for the maleic anhydride. So, so then we expect this rate to be measured, and we are working on that. So there are various challenges for these measurements. So one of them to be fully quantitative. So one of them is the rheology of the mixture, because this Diels Alder reaction may happen in one section of the reaction jar that is not accessible to the Raman laser. So we wanted to find an additive that could modify the rheology of the reaction so that the laser or the section that is of the jar that is illuminated by the laser would always have the same a rather uniform mixture of the reactants and the product. So then we try various additives, and we found Triton X100 as a dispersant or a non-ionic surfactant to form like a viscous paste in which we don't know if the reactants are dissolved or suspended, but the rheology is rather it can be uniform. It not always is uniform, but it can be uniform for for these measurements to happen. So most of that signal is actually the acrylic. So, and the acrylic has to be subtracted in a reliable manner. Has to be scaled and subtracted from each of the hundreds of spectra that can form one kinetic run. So this is another challenge, and we can see that the signal of the reactants, so this one is the reactant and this one is the other reactant, is just is a small fraction of what we measure, and this one is the product, and they have to be separated at different wave numbers. So this is a sequential number that we use for the analysis of the data, but it has an equivalent wave number there. And in theory, the quantitative Raman spectroscopy is not done with the peak heights, it's done with the integrated intensities of the peaks. So which would mean that we have to integrate the signal between certain wave numbers, and we have not done that yet. So then this is a 2D graph of the Raman peak height intensity versus the Raman shift. Here we have wave numbers, and we can see that here we have the signal of the product that starts to be more appear at around 5 minutes of reaction, and then it keeps increasing. And this one is the signal of the dimethylbutadiene. So we see here too that is decreasing. And this one is the signal of the maleic anhydride that is also decreasing. So then our for quantitative Raman spectroscopy, then we would need to work with the integrated intensities of those peaks. But typically people has published with the peak heights, not with the integrated intensities. So we are kind of testing those two approaches. So far, then, as I said before, this non-ionic surfactant Triton X100 allowed the measurement of these kinetic profiles. So the red one is the dimethylbutadiene, and the yellow one is the maleic anhydride, and the blue one is the product. And so it looks like the decrease in intensity or increase is there is no large changes. So it seems that the rheology is sufficiently good. The mixture is rather homogeneous for around the section that is illuminated by the Raman laser and detected by the probe. So this one is Triton X100. We don't know yet whether the reactants are dissolved and they form, we have like micelles of these, or if they are dissolved in this substance, or they are suspended, we don't know. And the product seems to, under the optical microscope, seems to form these needles and be suspended at the end of the reaction. So, but we are fitting these profiles. Right now, they are not second order overall as we would expect. So I just want to do more experiments before saying a lot more. So in conclusion, we have shown that liquids additives can lead to different ionic or neutral charge transfer complex polymorphs of tetrathiafulvalene according to their polarity or their electric constant, and sometimes according to their protic character. So they may have a role in the mechanism for the full or partial ionization of those donor and acceptor molecules. different polymorphs of the reactant tetrathiafulvalene can lead to different polymorphs of the product, and that is something that is not often seen in literature, in publications. So amorphous mechanochemical products then does not mean a physical mixture of the reactants, and they can have poorly crystalline seeds of the product that, upon exposure to vapors of solvents, can produce the rapid recrystallization of the products from the recrystallization of the products. And liquid additives such as Triton X100 can also modify the rheology of mechanochemical reactions, and this may be useful to monitor those reactions under homogeneous conditions by in situ Raman spectroscopy. So this is all what I have to say, and I am grateful for funding to NSF, to the Air Force Office for Scientific Research, partial funding to the International Center for Diffraction Data, and I'm also grateful for the use of the National Synchrotron Light Source and the BT1 high-resolution neutron powder diffractometer at NIST. So that's all, and just provide some questions.
We have a few questions from the audience. Let's start with this one. Have you attempted any reactive aging on these systems? And if so, what did you see?
I have not attempted aging. Basically, I guess I'm not 100% sure. I have exposed these reactants to vapors of those solvents, but without grinding. So in that case, after days of exposure to solvents, we obtain the products, the charge transfer complexes as products, but it takes much longer days.
Thank you. All right. A wide range of solvents was used for many of the experiments, such as the synthesis of TTFCAH2. The solvents were grouped according to their polarity, but excuse me, I'm sorry, I'm struggling here. But there was a large difference in viscosity as well. For example, DMSO, high, hexane, low. Do you know of any consistent effect of the solvent viscosity on polymorph formation?
Not that I know, and it's something that I have not explored, but if the green tetrathiafulvalene formation is under diffusional control, a larger solvent viscosity should de-accelerate that reaction, should make it slower. It's something for me to test in new experiments.
Thank you. Here's another related question. Doesn't adding a liquid help with the Raman tracking change the kinetics of the process compared to neat reaction?
I think it helps because the neat reaction, at least the Diels Alder reaction, may happen. The reactants and products may clump together. They may agglomerate in a region of the jar that is not illuminated by the laser. So the full reaction could happen, and we could not be able to actually see the products, and not like an homogeneous variation of the intensity, because it would be happening far away from the region illuminated by the laser. So for that, the additive was useful.
All right, great. Let's do one more and I'll pick a general question here. All right, a very general question. For the first two parts of your talk, why is mechanochemistry of interest? For example, is mechanochemistry much more efficient in producing those products than solvothermal?
Well, mechanochemistry would lead to one or the other polymorph by using a particular liquid additive in sufficient large quantities, at least from the mortar and pestle that I use at that time. in many cases, and sometimes from solution, one can obtain mixtures of those. And the people that studied these compounds a long time ago could not grow crystals sufficiently large of the black form to solve the structure from single crystal diffraction. So then the structure remained not determined until we could prepare the form pure by mechanochemistry and solve the structure from powder diffraction data.
All right. Well, thank you very much again for your presentation. It's our pleasure to host you.
Thanks.
Thank you, Dr. Pagola, for that outstanding presentation, and thank you so much for joining us as part of the CMCC Mechanochemistry discussions. Just a reminder that the YouTube site has all of our previous seminar series available to watch at any time. We also have a great group of speakers planned for the upcoming months, and we hope that you will join us for all of them. Thank you again.